Hi everybody, this is Mr. Nolan, and what I would like to do in this video is to really quickly go through the antibiotics web lab that we did a week or so ago, and kind of talk about how it fits into our resistance model. So in class we named the resistance model after certain students, but of course that's different in every class, so we can't do that in this video. But our goal here really is to be able to interpret the results of the antibiotics web lab and to use the results to develop a resistance model. So here's really what I'm talking about. If I show you here, this is the web lab that I'm really talking about. So you should recognize this. You should kind of see what it was that we did, and I can even zoom in a little so it's easier to see here. So our goal is to use this lab in order to explain how does our model work. So in looking at this lab, I'll skip all of the background explanation, but you might remember that you can control how many bacteria we start with. You can control whether they reproduce and how often. You can control the dosage of antibiotics and how frequently and everything. And it graphs it, and it also shows you over here what that population looks like. So what I have is I have 10 of each bacteria here, and I'm telling them, yes, reproduce every half hour, and I'm going to dose them at 75 milligrams, skip no doses. I'm just trying to show you a scenario that is basically guaranteed to give us the results that we want. So I'm going to go ahead and run this simulator, and we'll see what happens. All right, here we go. So that's our first wave of antibiotics, those little tiny dots that are going across the screen, and our bacteria are reproducing. So there's a number of factors at play in here. There's not just the bacteria reproducing, there's also the antibiotic is killing some of the bacteria. So I'm going to go ahead and pause our simulator, and we're going to look at the picture and look at our graph and try to interpret what is it that we're really seeing here. So you can see on the screen that our infection is not going away. So we see there's actually several of each color. We see that there's some brown here, there's actually a significant amount of red, there's some patches of green, but the one that's really outnumbering everybody else are these purple ones. And if we look over here at our graph, and I'll zoom in some more so we can get an even better look at our graph. So if we look here, we can see, well, isn't this interesting? We can see that as the simulation has run, and as we have dosed our population of bacteria over and over, we find that the brown, green, and red are doing the worst, right? They're actually all pretty low. The purple has come out in the very highest percentage. So almost half of our population is purple, and then the rest are a mix of the red, green, and brown. So what is this supposed to mean? And what do we actually draw from this? That the purple are way out competing these other bacteria. Well, if we take a really close look at them, and we can zoom in even more, if I sort of pull over here, if we take a really close look at these, what we notice, if we look at let's say look at our red bacteria, these ones, if you look really closely at one red bacteria, you can see there's a lot of holes in that membrane. There's actually 1, 2, 3, 4, 5, 6. There's all these holes. And the red actually didn't do that well. They did a little bit better than the green and brown, but they still didn't do very well. Over here, the brown have 5 holes, and they didn't do very well either. The green have 4 holes. And then finally, if we look at the purple, those have only 3 holes. 1, 2, 3, just 3 little holes. And those purple bacteria way outcompeted everybody else. And so the important thing to draw from this is that it's those purple bacteria with the fewer holes that are actually going to do better in the simulated environment. They're actually going to reproduce a lot faster. It appears as though the antibiotics don't really kill those bacteria that only have 3 holes, or the 3 pores. The fewer pores that the bacteria has, the better for the bacteria. Those bacteria are more likely to reproduce. And we saw that happen over here. The ones with the many pores didn't do very well. The ones with the fewest pores, the purple ones, actually did really well. They did better than everybody else. So here's what we are supposed to get from that. Here's what I want you to sort of draw to conclude from that simulation. We want to be able to interpret those results and use the results to develop a resistance model. So what I'm going to do is go over to the board over here, and we're going to try to draw a model together for how do bacteria become resistant. And this is basically sort of the point of the whole unit, is to be able to figure out, how does this happen? How do the bacteria become resistant? So I'm going to go over to the board and I'm going to draw a few things. Let's go ahead and start off by drawing the non-resistant population, because that's what we start with. Okay, what I've drawn here is our population of non-resistant bacteria. That's what we started with, right? We would not say that we started with resistant bacteria. However, not all the bacteria were the same. When we looked over here, remember, we didn't start with all the same color. We actually started with kind of a mix, and these different bacteria are all a little bit different. I've tried to kind of color code these according to the model. So we've got our red bacteria with 6 pores, we've got our green bacteria with 4 pores, and we have a couple of bacteria with only 2 or 3 pores, and those I've drawn blue because it's the closest to purple. Now, the first thing that happens in our model is that we hit them with antibiotics. So I'm going to go ahead and show one way that we could model this to show that the bacteria have been hit with antibiotics. So basically what I've done here, in order to show what is happening in our simulator, I'm drawing an arrow to show the passage of time, and I've written antibiotics by it. And the X's are showing that those bacteria with the more holes, the more pores, these other colors here, are not, they don't respond very well to that antibiotic. But the purple ones tended to survive. They were much more likely to survive. So those survivors, I'm going to go ahead and draw those over here. Okay, so here we have the antibiotics killing off all the weak ones, all the weak bacteria, and here's our survivors. Now before we continue our model, it's worth asking, what is special about them? Why are they the survivors? What's special? And that's because they have a special gene. They have the resistance gene. And that's why they have fewer pores. So these bacteria have a gene that gives them fewer pores, and as a result, they are basically resistant, or they're more resistant than the other bacteria are. So that's worth kind of adding in here, that those ones that survived, it's not just because they were lucky, it's not a coincidence. They survived because they had a different gene. They had a mutation. They had a mutant gene that these other bacteria don't have. Okay, so I've included this sort of labeling that these blue ones that have fewer pores, they have a mutant gene. They have a gene that has mutated. Now, anytime you hear the term mutant or mutation, all that means is that the DNA has changed. It's a fancy way to say that there was a change in the DNA. So these mutant bacteria have different DNA than the normal ones that died, right? These ones died. So now what's going to happen over time? As these survivors sit there in your body or on a petri dish, what are they going to do? You are absolutely right. They are going to reproduce. So let's go ahead and illustrate that here in our model. Okay, so we've got reproduction here. What we're going to do now is we're going to show, now that those survivors are reproducing, how many are there going to be after several rounds of reproduction. If they reproduce one time, you can maybe do the math in your head. I sure hope you can. If I have 2 cells here, if I have 2 mutant bacteria, and they reproduce one time, how many am I going to get over here? You just multiply it by 2. So I should end up with 4 cells over here after some reproduction. Okay, so there's my new population after 1 round of reproduction. It has doubled, right? That's how the math works when you do reproduction with bacteria. So now that those survivors have reproduced, essentially what we have here, this is the resistant bacteria. Now, I'm oversimplifying what we saw in the web lab. It's I'm trying to simplify it so we can see what are the important results that matter. Here they are. We end up with more mutant bacteria, those survivors, those resistant bacteria. And so this is the antibiotic resistance model. Here it is. This is it right here. We just have to show those non-resistant, those weak bacteria dying because they have the normal gene, and the mutant gene survives, it reproduces, and now we have more of them. So this is the population of resistant bacteria. There they are. And it's worth keeping in mind, they all still have the mutant gene, because as these survivors reproduce, their offspring are inheriting the gene. Bacteria, just like us, bacteria inherit DNA from their parents, and so they inherit that mutant gene, and so now the resistant bacteria has a lot more mutant genes in it than when they started. Okay, so this really is our antibiotic resistance model. Here it is. So if when in doubt, maybe sketch something like this. But this also helps us to explain lots of stuff that we see in the natural world, and we're going to move forward after this unit into looking at plenty of other examples in which organisms can change over time in exactly the same mechanism. This is a mechanism by which this happens. So I hope that this makes sense to you. If it doesn't, try sketching it on your own, maybe practice a little bit, and make sure that you understand how do reproduction, antibiotics, the surviving bacteria, how does all these things factor together, the mutant gene, to get us from non-resistant bacteria over to resistant bacteria. It takes a couple of steps in order to do that. But I hope that that's clear to you, and that you've met our goal of being able to interpret the results of the antibiotic web lab in order to and use the results to develop a resistance model. So I hope that you can do that at this point.